Hello guys, this is Sai and welcome back to our channel Lovely Pharmacy. Today our subject is about herbal drug technology. Herbal drug technology or HDT. Okay, today we go through with the basic concepts or basic definitions in HDT. What are these basic definitions in HDT? So first herbal medicine plant, next herbal medicine product and herbal drug preparation and source of herbs. Okay, the word herb is derived from the Latin word of herba. Latin word of herba, which means a grass or green star, which means a grass or green star, and the entire plant or a part of plant is called herb. అంటే ఒక ప్లాంట్లో ఫర్ ఎగ్జాంపుల్ ఇది ప్లాంట్ అనుకో, దీంట్లో ఒక చిన్న పార్ట్ ఒక చిన్న బ్రాంచ్ ఆర్ స్టెమ్ అనేది హెర్బ్, లేదా ఇది ఎంటైర్ ప్లాంట్. సో ఇది కాల్ ఈ చిన్న పార్ట్లో మెడిసినల్ వాల్యూ ఉంటే దాన్ని హెర్బ్ అంటారు. ఆర్ ఎల్స్ ఇన్ సమ్ కేసెస్ ద హోల్ ప్లాంట్ కన్సిస్ట్ మెడిసినల్ వాల్యూ. ఓకే, దట్ ఈస్ కాల్డ్ హెర్బ్. నెక్స్ట్ herbal medicine or herbal medicine plant. Next herbal medicine plant. The herbal medicine is also described as herbalism or phytomedicine Herbal medicine is called herbalism or phytomedicine, which consists of the plant which consists of a therapeutic action. For example, this is a herbal plant. This plant consists of a therapeutic value or therapeutic action. But we cannot say this is completely a good plant. This is 100% this plant consists 100% therapeutic value. We cannot say like that, because this herbal plant also consists few chemical reactions or few chemical side effects or chemical toxicological effects. For example, this herbal plant consists of 95% of herbal value, therapeutic value, may contain or may not contain 5% of chemical value, may contain or may not contain 5% of chemical value. So side effects, the side effects are also there for herbal medicine plant, but those side effects are in less. Next, herbal drug preparation, herbal medicine product. This herbal medicine product, the herbal medicine product is obtained or processed by the herb. The herbal medicine product is obtained by the herb by few methods like infusion, extraction, decoction. By these three infusion, extraction, decoction, by these three various methods, the herbal medicine products are prepared by the herb or called herbal medicine product. Now for example, this is a herbal medicine product. This product, in second point we discussed it also contains some 5% of chemical effects. Now in third step, the herbal medicine product, when it is extracted by the infusion, extraction or decoction method, these 5% chemical side effects are removed in this process. In this any one process is used to remove this 5% of side effects. Now this is completely now the herbal medicine plant becomes completely 100% medicinal plant. 100% medicinal drug plant. Medicinal drug. The medicinal drug is obtained by in this. Next, herbal drug preparation. These herbal drug preparations are described as a cuminated or powdered or herbal powdered form. Cuminated, these herbal drug preparation are extracted or are done by cuminated or herbal powdered form, which extracts essential oils, essential oils like fatty oils, tinctures, juices are done here by using purification by using purification, infusion, and fermentation, by using purification, infusion, fermentation, and hybridization. By using purification, infusion, and fermentation, by using these three process. Next, decoction. By using purification, infusion, fermentation, and decoction, by using these four different process, by using these four different methods, the herbal drug preparations are done. Next, we go through it. Source of herbs. Source of herbs is classified into 5 different types: wild source, cultivated source, plant tissue culture, hybridization, and polyplied mutation. So first we go through with wild source. Basically, the wild source is obtained from the uh, obtained from the forest plain, forest plains and river banks. These wild sources obtained from forest, plains, and river banks. Forest means we know that we know where the huge number of plants are grown are called forest. Next, plains. Plains is nothing but a open area where the plants are easily able to grow. Where the plants are easily to grow is called plains. Next, river banks. River banks also we can know. In river banks, mostly the mangrove trees will be grown, and a few other plants are also grown near the river banks. These the specialty of these wild resources. These do not require any human care. And these produce different varieties. Different varieties. And these go faster, and the wild plants also grow faster, and these contain excipients, and these wild sources also contain one or more active ingredients. So wild source, these wild sources are obtained from the forest, plains, and river banks. So these do not require any human care. These grow or these produce new or different varieties of plants, and these consist of excipients also, and it consists of one or more active ingredients. Next, cultivated source. The herbs are cultivated. By using modern and scientific techniques. Cultivated. So this is cultivated by using modern and scientific techniques. The herbs are cultivated. Here, what are the process? First is tissue culture. Hybridization, and mutation. By using these three different by using these three different methods: tissue culture, hybridization, mutations, the herbs will be cultivated in the cultivated process. And the advantage of these cultivated processing is, we can get advantages are purity and more yield. And applications like, and we can use applications of modern and scientific technology, get purity, we can get purity of the plant and more yield, and applications of modern and scientific technologies are used in this, or in cultivated. Next, plant tissue culture. Third one, plant tissue culture, PTC, plant tissue culture. The plant tissue culture is an in vitro technique. The plant tissue culture is an in vitro technique. New plants with the new different types of plants are able to produce in the tissue culture, in the plant tissue culture. For example, plant tissue culture is defined as a producing of new different types of plants by using a plant stem, or branch or leaf, or by seed. By using these parts of plants, in nutrient media or essential nutrient, by using nutrient media, we can grow the plant tissue culture, or we can generate the plant tissue culture in the nutrient media. See, for example, this is a beaker, and this is a this is a beaker or any type of container. This is a nutrient which is required to grow the plant tissue culture. These may be stem, or maybe the branch or the leaf or the seed of the parent plant is taken, and it is transferred into the nutrient medium, and within the few days the plant starts growing, and it is also grown to another parent cell, another parent plant, not cell, so another parent plant. This is called plant tissue culture. I mean, obtaining a leaf or stem or branch from the parent plant and able to produce a new plant is called plant tissue culture. Next advantage of this plant tissue culture is we can produce exact number of plants. And disease-free plant. Next, genetically modified plants are also able to produce. First thing, disease-free, genetically modified plant. Desired number of plants are copies. These are number of plants are copies are able to produce. Okay, these are disease plants, genetically modified plants. Next, these are number of plants are able to produce in the plant tissue culture. Next, hybridization. Next, fourth one, hybridization. Hybridization is defined as a producing a new variety of plants by crossing two different types of species or varieties in plants. అంటే ఒక టూ డిఫరెంట్ టైప్స్ ఆఫ్ ప్లాంట్, అంటే ఇప్పుడు ఫర్ ఎగ్జాంపుల్ ఇప్పుడు ఒక మెయిస్ ప్లాంట్ ఉంది. ఆ మెయిస్ ప్లాంట్ని సేమ్ స్పీషీస్, ఒక మెయిస్ ప్లాంట్ ఈజ్ స్పీషీస్, అండ్ ఇందులో వెరైటీ ఇందులో ఇంకో డిఫరెంట్ వెరైటీ ఆఫ్ మెయిస్ ప్లాంట్ తీసుకొని ఈ టూ క్రాస్ అంటే ఒక సేమ్ స్పీషీస్ బట్ డిఫరెంట్ వెరైటీ తోటి క్రాస్ చేస్తే నీకు ఇంకొక న్యూ డిఫరెంట్ వెరైటీ ఆఫ్ మెయిస్ ప్లాంట్ అనేది ప్రొడ్యూస్ అవుతుంది. దట్ ఈస్ కాల్డ్ హైబ్రిడైజేషన్. సో హియర్ ద మెయిస్ ప్లాంట్ ఈజ్ జస్ట్ సింపుల్ example I use it here to understand you. So hybridization is also, once again I am telling, hybridization is defined as a producing of new variety of plant by crossing two or more plants is called hybridization. Okay, got it? This hybridization is subdivided into four types. Hybridization is intervarietal, intravariatial, intergenic, and interspecific. So hybridization lo intervarieties and intervarieties and intravarieties and intergenic and interspecific. These are the four subdividing hybridization. Next, polyploid mutation. Polyploid mutation is described as having multiple chromosome sets multiple chromosome sets. Multiple chromosome sets, and begin introduced by some chemicals, like colchicine, colchicine, to increase the yield value. To increase the yield value of the plant is called polypeptide mutation. So once again I'm telling, polypeptide mutation is described as having a multiple chromosome sets. అంటే polypeptide mutations లో దీనికి ఒక సెట్ ఆఫ్ క్రోమోజోమ్ ఉండదు, మల్టిపుల్ like two chromosome sets are having in the polypeptide mutations, and in this the colchicine chemical is induced in this. The colchicine enzyme, the colchicine chemical is induced in the plants to increase the yield value, mainly banana. Examples: banana, wheat and strawberry. For these plants, mostly the colchicine chemical is used to increase the yield value. Is called the colchicine chemical is used in these plants, banana, wheat and strawberry, to increase the yield value. So it's called polypeptide mutation. Okay, I hope you guys understand what this mean by herb and medicinal plant and medicinal product and drug preparation and source of drug. So okay guys, in next class we will discuss about selection, identification and authentication of herbal plants. Okay, so not to miss the next video, subscribe down. Yeah, okay, thank you guys.